This paper, just published in Nature Neuroscience, offers something the autism neuroimaging field has long been searching for: empirical, biologically grounded subtypes of autism, not defined by behavior, but by brain connectivity and its underlying molecular pathways. Autism is heterogeneous. Individuals differ enormously in symptoms, genetics, and brain imaging profiles. The prevailing assumption has been that this phenotypic variability reflects distinct underlying pathobiologies, but direct evidence for that link has been absent. Genetic stratification only works in the roughly 20% of individuals who carry identifiable rare variants. So, the authors ask, can we use fMRI connectivity itself to decode biologically distinct subtypes? Their approach is elegant, and it starts in rodents. They assembled resting state fMRI data from 20 different genetic mouse models of autism, spanning synaptic, transcriptional, immune, and copy number variant etiologies. When they looked at voxel-wise connectivity differences, mutant versus wild type, across all 20 lines, two dominant patterns emerged: a hypoconnectivity cluster comprising 11 models and a hyperconnectivity cluster comprising 9. These two subtypes showed negligible spatial overlap and distinct large-scale network signatures: hypoconnectivity in salience and default mode networks, hyperconnectivity in limbic and subcortical systems. The critical step was asking what biology separates these clusters. Using protein-protein interactomes built from the seed genes of each subtype, and then gene ontology analyses, they found a clean dissociation. The hypoconnectivity subtype was strongly enriched for synaptic pathways, synaptic transmission, SYNGO, MAPK, WNT signaling. The hyperconnectivity subtype, by contrast, showed no synaptic enrichment, but strong enrichment for immune and transcriptional mechanisms, cytokine signaling, innate and adaptive immune response, chromatin remodeling. The biology maps onto the fMRI. Guided by those rodent disconnectivity maps, the authors then turned to a large multi-center human dataset. 940 individuals with idiopathic autism and over 1,000 neurotypical controls from 38 albide and CMI collections. Using a cross-species regional decoding approach, they identified analogous hypoconnectivity and hyperconnectivity subtypes in humans. These were highly replicable across a held-out 21% replication cohort, dice coefficients of 0.74 and 0.96 respectively. Together, the two subtypes accounted for about 25% of the autism sample. Critically, human gene decoding of these subtypes recapitulated the same dissociation: synaptic genes for hypoconnectivity, immune genes for hyperconnectivity. The hyperconnectivity subtype also showed higher autism severity on social affect scores. What does this mean? This is the first cross-species empirical framework that links fMRI connectivity subtypes directly to dissociable biological pathways. It raises the possibility of biologically informed stratification, which matters enormously for treatment trials. That said, the subtypes currently account for only 25% of the autism fMRI data examined. The majority remain unsubtyped. The approach also relies on evolutionarily conserved brain regions, which introduces its own translation constraints. But as a proof of concept that brain connectivity heterogeneity encodes real biological signal, this is a meaningful advance.